am Micaela Sosa. I work with Vanessa Galassi and Mario Del Popolo in the Biophysics and Soft Matter group at Senum Cuyo. The job I'll present you is about partition and optical response of a voltage-sensitive dye in polarized lipid membrane from a computational approach. Have you heard about voltage-sensitive dyes? These molecules are used to measure membrane potential in vitro or in cells due to their capability to change their optical properties when they are exposed to an electric field. I am going to talk to you about one of them, indocyanine green. This dye has reported sensitivity to voltage and is attractive because it's the only fluorescent dye approved by the Food and Drug Administration for clinical use. Additionally, its fluorescence absorption and emission is in the near-infrared region of the spectrum, where light penetration through tissue is maximal. Consequently, with this dye, images of electrical activity could be generated without requiring highly invasive procedures. We have developed a computational study to investigate which are the molecular bases involved in the differential optical response of ICG to changes in membrane potential. Our objective was to evaluate how much the configuration of molecules contribute to the differential optical response and how much the electrochromic effect contribute to it. The simulation box was composed of a double bilayer in order to establish two separated compartments. Membrane polarization was obtained a plane and ion imbalance in the two compartments. To obtain energetic and configurational contribution, we first calculated the profile of the energy of the insertion process of ICG from bulk solution to a bilayer of palmitoyl oleiphosphatidylcholine through umbrella sampling method. The harmonic constraint was applied along the distance between the dye and the cis bilayer center of mass. Here you can see how both systems have a similar affinity to membrane, thus the insertion of ICG is equally favorable for both of them. After that, we have calculated localization of ICG in a simulation beginning from the energy minimum configuration and releasing all bias. According to the minimum location in the free energy profile, we have obtained that it doesn't depend on membrane polarization state. Both systems show a very similar depth for the dye insertion in the membrane. Then we calculated the tilting dye ICG presented in the most favorable localization, observing that ICG had a mean angle around 100 degrees in both systems, but with great variability in polarized systems. To evaluate variability in the conformation of ICG in the bound state, we grouped similar conformers of unbiased trajectories. We set an root mean square deviation criterion in order to obtain the 80% of conformation in the first 20 clusters for both systems. Then we calculated absorption spectrum for the mean conformation of each cluster. Here, I show you the weight sum of conformers spectra corresponding to polarized and unpolarized systems obtained by time-dependent DFT calculations. With our calculation, we have obtained a redshift around 15 nanometers for the polarized system regarding the unpolarized system, which is in good agreement with experimental results. However, we couldn't reproduce experimental wavelength, which is around 780 nanometers. Finally, looking to improve agreement with experimental wavelength, we have performed TD-DFT calculations with different functionals and basis set to the dye-optimized geometry, but we have not improved the agreement with the expected wavelength. To conclude, we have obtained a mean tilting angle very similar for both systems, around 100 degrees, but with greater variability in the polarized system. We have obtained a bathochromic shift of 15 nanometers in absorption spectrum upon polarization. Each shift is in the order of that reported experimentally. And as perspectives, we are going to continue looking for an appropriate functional and basis set which allows us to reproduce mean absorption. We want to acknowledge Team Fellowship, CONICET, and Agency for fundings. Thank you.